Namaskar, this is Vaccines Part 2. In previous lecture, we have read about conventional vaccines. Now, novel approach for various vaccines and vaccination. Novel approach, emergence of new technologies now allow new advances in vaccine development, including design of new vaccine molecules by genetic engineering, vaccine against herpes simplex virus, macromolecular purified vaccines, recombinant vaccines against foot and mouth disease, live recombinant vaccines against Vibrio cholerae, live recombinant vector vaccine, synthetic peptide vaccine, recombinant naked DNA vaccine, and anti-idiotype vaccines and malaria vaccine. Design of new vaccine molecules by genetic engineering. Through genetic engineering, it is possible to modify the genes of a pathogen so it becomes effectively and reversibly attenuated. A vaccine has been developed against herpes virus that cause the disease pseudobarbies in pigs. The thymidine kinase gene has been removed from the virus. In the absence of gene thymidine kinase, it not synthesized by the virus that stop it from replicating. The virus is thus disabilized and could no longer cause disease. This genetically modified virus without thymidine kinase gene is used as vaccine. This virus, although invades nervous system but does not replicate, hence cannot cause disease. The other genetic method for rendering an organism disabled is gene assortment, as exemplified in rotavirus and influenza virus. The virulent genes may also be deleted, as in Vibrio cholerae, in which toxin genes may be removed. In another mechanism, missense genes is inserted, which cause generation of temperature sensitive mutants. Vaccine against herpes simplex virus. Herpes simplex virus is the causative agent of cold sores, cancer, sexually transmitted diseases, encephalitis, and painful eye infection. HSV persists in human host by hiding from immune system in sensory neurons. First is infect epithelial cell, then spread in neuron. The HSV remains dormant during onset of host immune response. It reactivates itself when the immune system subsides. Recently, herpes simplex virus has emerged as most notorious pathogenic virus. It occurs in two strains known as type 1 and type 2. The infection of disease occur by direct contact with epithelial cells tissue through blisters of patients. The infection of type 2 occurs by sexual contact with infected individual. The key for the development of an effective and subunit vaccine is to characterize the antigenic component of the pathogen that could elicit maximum antibody reaction against the virus. The HSV virus envelope protein glycoprotein D, also known as glycoprotein D, is GD protein, is injected into mice which induce specific immunoglobulin to neutralize HSV1. The gene encoding for protein HSV1 GD has been isolated and cloned into Chinese hamster ovary, where it is express itself. The mammalian system permits foreign protein to be glycosylated. The GD protein chain binds to plasma membrane of the chick hamster cells, Chinese hamster's ovary cells, since it is difficult to purify the membrane-bound enzyme. Hence, GD genes was altered to remove the nucleotides of the gene expressing the C-terminal transmembrane domain binding to the plasma membrane. The modified gene construct was transferred to Chinese hamster ovary cell, where the gene product did not bind to plasma membrane and was secreted into the external medium. The modified glycoprotein D was found to be an effective vaccine against HSV type 1, HSV type 2 viruses. Macromolecular purified vaccines. Sometimes attenuated or killed vaccines induce adverse effect on host. This risk can be avoided by designing purified macromolecules such as bacterial capsular walls. Purified macromolecular vaccine against pneumococcal bacteria Streptococcus pneumoniae consists of 23 antigenically different capsule polysaccharide. The above bacteria cause pneumonia. The vaccine induces the formation of host protective antibodies and is recommended for all infants. The vaccine against the bacteria Neisseria meningitis that cause meningitis is composed of purified capsular polysaccharide. Although the bacterial polysaccharide vaccines activates B cells, but it does not induce antibody class switching and memory B cell production. This vaccine also fails to activate CD4 cells, but it induces production of IgA secreting plasmocytes in man, following by subcutaneous immunization. It also activates proliferation of IgA secreting specific memory vessels which are produced by bacterial antigen at mucosal surface. The polysaccharide vaccine is also used of Salmonella typhi, Haemophilus influenzae, Escherichia coli, and Klebsiella pneumoniae. The capsule determines both pathogenicity and antigenicity of the organism. Tetanus diphtheria produces exotoxin. This exotoxin, after purification and inactivation with formaldehyde, constitute toxide. Vaccination with toxide induces anti-toxide antibodies which also bind with toxin and neutralize its effects. The vaccine is called DPT, diphtheria, pertussis, and tetanus. The DPT vaccine has been superseded by DAPT, which pertussis bacteria is replaced by acellular component of pertussis. The capsule especially contains modified inactivated form of pertussis antigens such as filamentous hemagglutinin, pertuxin, and fimbrial antigens. The antibacterial vaccines are made from whole bacteria. Their secretors toxin, especially capsular polysaccharides.